Thank you all for joining us for this special edition. My name is Eli Soto. I'm a board member of the Pheo Alliance, but also, like many of you guys on this webinar, looking for answers and having lots of questions in your mind. I was once like you. I was a patient. I had a metastatic pheochromocytoma for who knows how long. They estimate based on the size and how much it spread, potentially over 10 years, but nobody really knows. Had I had access to these webinars or even known about it back then, this would have given me wealth of knowledge because they get some of the best and brightest minds from around the world to educate the general public and other clinicians about this disease. So this is a really good treat that we're in for today. But I will say, for myself, after being in remission, it's like a whole new world. It no longer feels like a lion is chasing me every day. The best word I can use is calm. Everything just feels calm. And I think many of you guys are looking for that. So hopefully today you'll figure out some more information that'll get you one step closer to that. So as we already mentioned, we're here with Dr. Rory Clifton-Bly, and I will properly introduce him in a moment. But first, I want to let you guys know that this program is brought to you by the Pheo Alliance, whose mission is to empower patients with pheochromocytomas or paragangliomas, their families, and medical professionals through advocacy, education, and a global community of support while helping to advance research that accelerates treatments and cures. And we'd like to give a special thanks to Progenics for making this webinar possible through a series of educational grants. A little bit of housekeeping. Our inaugural Pheo Para Awareness Week is coming up August 23rd through the 29th, and our motto is Pheo Para Fearless, spelled with a PH. So we need your help to participate, so please go on pheopara.org for more information. There's an upcoming webinar at the end of this month on August 29th, covering Pheo, Para, and GIST with Dr. Siklik. So a way that you can go ahead and keep on top of this is just sign up for our e-news or follow us on social at Pheo Para, whether it be on Facebook or on Instagram. So for today's agenda, for about 25 minutes, Dr. Clifton-Bly will be presenting. Then we'll be taking Q&A from all of you guys. Some of the questions have already been submitted beforehand, but we'll also look for your questions that are going to be submitted online through the chat function. Many of you guys will ask variations of the same question. That's very common. So listen very carefully to all the questions and answers because they're very similar sometimes. Questions that are less case specific are appreciated. And to that note, I'll go ahead and do a little disclaimer that the information presented on this webinar is for educational purposes only and should not substitute the advice of your doctor or doctors and medical team, because they have an in-depth knowledge of your particular case and current situation. So, with all that said, I'd like to go ahead and introduce Dr. Clifton-Bly. Dr. Clifton-Bly is the head of the Department of Endocrinology at Royal North Shore Hospital and co-joint professor in medicine at the University of Sydney. He completed a PhD in the genetics of thyroid disorders at the University of Cambridge and now supervises dual research programs, one of which focus on the genetics of endocrine neoplasms and the other on metabolic bone disease. The Cancer Genetics Unit studies the molecular basis of thyroid cancer, pheochromocytoma, paraganglioma syndromes, adrenal cancer, and pituitary neoplasms, and he maintains strong involvement in the teaching and mentoring of young physicians. Quite a busy man. So, without further ado, I'd like to turn it over to Dr. Clifton-Bly to go ahead and share some knowledge with us.
Thank you so much. It's a real pleasure to be with you, and greetings from chilly Sydney, where it's 15 degrees Celsius. I'm not sure what that is in Fahrenheit, but I also want to acknowledge that these have been extraordinarily difficult times for us all across the globe, and my thoughts and prayers are with you and your families as we navigate these uncertain times amidst the coronavirus. So we could spend days talking about paraganglioma research, so I make no apologies for cherry picking some topics that I thought would be of interest to you and which I hope can promote some lively discussion. Just a few disclaimers. I don't think any of those disclosures have anything to do with what I'm going to talk to you about today. So I want to talk about how we understand the genesis of pheochromocytoma by understanding genetics. And then I want to talk about the development of targeted therapies. And then I want to talk in the third section about prevention of metastatic disease. And I presented to the Pheo Para Alliance in Syracuse last year. It was a terrific meeting. I was honored to be there. And some of this overlaps with what I spoke about at that meeting a little more than 12 months ago. So determining the drivers of pheochromocytoma development is essentially understanding the genetic code for Pheo. So we can look at the genetics by sequencing DNA, and I'll speak to you a bit about that. And there's a new understanding about the drivers of Pheo development, which a new science called metabolomics, and I'll speak to you a little bit about that as well. So pheochromocytoma and paraganglioma is the case par excellence in human cancers for being heritable. Nearly 40% of people with a pheochromocytoma or paraganglioma will be carrying a heritable susceptibility gene. And there are now at least 12 of those genes, and many of the genes will be known to you all. Some of you may be carrying one of the genetic abnormalities. But Patricia Dahier 15 years ago recognized in a seminal paper in PLOS Genetics that the genes were clustered into two broad categories. There were the cluster 1 genes, which include SDH genes and VHL, and there were the cluster 2 genes, which included RET and NF1. And over the last 15 years, those gene lists have grown, and it looks a bit like alphabet soup at the moment. Lauren Fishbein added a third cluster in her paper 3 years ago from the TCGA consortium. But the purpose of showing this alphabet soup to you is that the gene clusters contain two broad categories of gene. You can have either a tumor suppressor gene, such as SDHB, wherein there's genetic susceptibility for cancer is related to loss of the gene. So these tumor suppressors in normal circumstances act to keep tumors at bay. And if you lose those crucial defenses, then tumors activate. And we call those genes tumor suppressor genes. But the other category of genes are called oncogenes. And these are genes whereby activation of that gene is responsible for determining the cancer program. And examples of oncogenes include the RET oncogene, which affects people with multiple endocrine neoplasia type 2. There's a few other oncogenes listed here, but the oncogene, the reason for highlighting oncogenes is there is a general principle in cancer medicine, and that is it's easier to make a therapy that inhibits something than it is to replace something that's missing. So when you look through cancer therapeutics, the vast majority of cancer drugs are inhibitors of some biologic process. And many of these inhibitors, particularly in this era of what we call personalized medicine, inhibitors are designed to specifically target some oncogene that is activated in an individual's cancer. And the oncogene I want to focus on today is EPAS1, and I'll come back to that in a slide's time. The reason I thought it would be interesting for you is that the Nobel Prize in Medicine last year was awarded to these three fellas, Greg Semenza, William Kaelin, and Peter Ratcliffe, for their discovery into how cells sense and adapt to oxygen availability. And you might say, what does that have to do with my pheochromocytoma? Well, it turns out that all three of these researchers have done research in pheochromocytoma. And in fact, pheochromocytoma was part of the understanding of oxygen sensing by the body. And so just to give you the potted summary of what was decades of elegant work from these three Nobel laureates, they essentially discovered how hypoxia-inducible factors, these are proteins, how these are regulated and how they regulate the body response to decreased oxygen levels. So I've drawn the protein HIF, hypoxia-inducible factor, and these little P's are phosphorylation sites. So HIF can be phosphorylated, and these are prolines, I should say, and these prolines are chemically modified by adding hydroxyl groups by an enzyme called proline hydroxylase. And that hydroxylated HIF is then recognized by the von Hippel-Lindau protein, and that then targets it for degradation. So under normal oxygen tension, HIFs are degraded. But if oxygen levels fall, it switches off the enzyme proline hydroxylase, and that allows HIF to activate target genes, which are very important in cancer biology. So this, as I say, was decades of work from the 3 Nobel laureates to understand how the body senses and responds to low oxygen tension. Now, it turns out that HIF comes in 2 different flavors. There's HIF1 and HIF2, and HIF2 is very important to pheochromocytoma and paraganglioma. The seminal discovery that HIF2 is important for pheochromocytoma was made by Carol Pachak and Zhengping Zhuang in 2012 in the New England Journal of Medicine, where they described a few patients with mutations in this gene associated with paraganglioma. We now know that mutations in this gene occur in the tumors of about 5 to 10% of all pheochromocytomas, so it's not an uncommon event. I should emphasize that this is not hereditary, so these mutations are within the tumor, but you can't pass these mutations on to your children. But nevertheless, it gave us an insight that this was potentially a target against which you could develop drugs that can help and benefit patients with pheochromocytoma. It turns out that not only can HIF2 be mutated, it's also generally overexpressed in pheochromocytomas, especially those cluster 1 pheos that I mentioned a moment ago. So the cluster 1 pheos are those associated with von Hippel-Lindau syndrome or the SDH syndromes, SDHB. So these cluster 1 pheos seem to express HIF2 at much higher levels than other pheochromocytomas. So as I say, in cancer medicine, it's easier to make an inhibitor than it is to fix something that's missing. So there's been a lot of interest in, can you target HIF2 inhibitors for the benefit of patients with cancer? And it was a long journey, but a company called Peloton has developed a couple of compounds, one of which it was published as a phase 1 trial for kidney cancer, where HIF2 is also very important. And they showed a prolonged response in at least one heavily pretreated patient. And this concept of using HIF2 inhibitor has been intensively studied by Rodrigo Toledo in Spain, but also Patricia Dahir in Texas. And I know that they're trying to escalate a clinical trial involving this Peloton compound in patients with pheochromocytoma. So the second topic that I want to talk about is the SDHB group of pheochromocytomas. SDHB is one of the commonest genetic causes, hereditary causes of pheochromocytoma. It accounts for about 10% of all patients with pheopara tumors. But it's very overrepresented in patients with metastatic disease. So of all metastatic pheochromocytomas, 40% will have SDHB mutations. But what we've realized for a while is that mutating SDHB just alone is not sufficient for progression of disease to metastases. So these, you can have SDHB mutations and a tumor, and it can progress in a very indolent fashion until something triggers it off to progress more rapidly. And as some of you are aware, we've been involved in a large international alliance, the American, Australian, Asian Adrenal Alliance, the A5 group. And one of the studies that A5 has supported is a study specifically addressing why patients with SDHB mutations are more likely to get malignant disease. And in this work, Richard Tottle, my friend and colleague at Melbourne, and Carol Pachak at the NIH, of course, have been leading this study, and I'm delighted to say it's very nearly at the point of publication. The headline data is that patients with SDHB mutations are more likely to develop malignant disease because they acquire secondary mutations in genes that activate telomerase. What's telomerase? It's an enzyme that keeps the end of our chromosomes from falling apart. So as we get older, these ends of chromosomes called telomeres shorten, and that's part of the natural aging process. And the same is true for cancer cells. As cancer cells divide, the telomeres will shorten, and if the telomeres get too short, the cancer cell will die. So one of the defense strategies a cancer cell has to keep dividing is to keep this end of the chromosome safe and protected. And that's what the enzyme telomerase does. Turns out that in patients with SDHB mutated paragangliomas, there's a mutation in telomerase that activates it in about 20% of SDHB related tumors. And there's another pathway that keeps telomeres safe in about 5% of these tumors. And there's this very strong association in these genetic events with metastatic disease. This discovery is, I think, absolutely vital for recognizing patients at risk for developing metastatic disease. It also gives us a glimpse of a new target that we might be able to develop therapies to inhibit. The missing link now that we're racing to discover is the mechanism by which succinate dehydrogenase mutations lead to telomere maintenance. And this is where we need to go back to our biochemistry knowledge. So succinate dehydrogenase is an enzyme in the mitochondria which converts succinate to fumarate. It's a biological process called the Krebs cycle. It's also part of the battery of the cell in what we call the electron transport chain that generates ATP. And so SDH functions as a dual purpose enzyme to convert succinate to fumarate in the Krebs cycle and to provide electrons that will generate ATP. So if you take out any of the SDH subunits, but here on the slide, SDHB, for example, you'll knock out the enzyme, and that will result in two very important consequences. Firstly, you get a rise in succinate because the enzyme is no longer able to change succinate to fumarate. So there's essentially a backlog, and the succinate increases. The second thing that happens is you lose the electron transport chain. So that means you've got a lot of free electrons floating around, and that generates reactive oxygen species. Now, we know some of the things that succinate and reactive oxygen species do, but this is a target area for research to understand the link between those two processes and the telomerase mutations that I mentioned to you just a moment ago. And we really don't know the steps in between here and here. And this is where a lot of research is being focused at the moment. Is it due to reactive oxygen species? Is it due to succinate? If we can work this piece of the puzzle out, then we're halfway there for developing new therapies that will help people with metastatic disease. Conceivably, it might be possible to lower succinate levels. Conceivably, it will be possible to target the reactive oxygen species. And I know there was a question from the audience about high-dose vitamin C. And megadose vitamin C was used in a trial by Carol Pachak, and this very much targets the reactive oxygen species. So, and the intravenous vitamin C is designed to mop up a reactive oxygen species. So that targets one part of this puzzle, and it had some very promising, but I would say initial and preliminary results, and the rest of the international community is now trying to work out just the magnitude of that potential benefit. Appears to be very little downsides to giving intravenous vitamin C, but time will tell to see if it really does fulfill its promise as an anti-cancer agent. So there are a lot of potential mechanisms that can join succinate to maintenance of telomeres, and so it's a bit like trying to work out a jigsaw puzzle, which piece goes where to make up the whole picture. And I just won't go into this detail because I think it'll be deadly dull for you all, but just suffice it to say that there's a lot of science that joins two simple dots, succinate or reactive oxygen species, with telomeres and keeping cancer chromosomes from falling apart. So if we can work out how those dots join together, then we'll be able to develop new cancer therapies. Some very interesting research. This is not my research. This is from Hans Gay from Florida, and a whole cast of international collaborators, and also from Carol Pachak, suggested that it wasn't just succinate that was abnormal in patients with SDHB pheochromocytoma. It comes to a concept of what we call metabolomics. Metabolomics is using mass spectrometry to study metabolites within cancer cells. And so one metabolite is succinate, but there are a whole lot of other metabolites that you can study using a technique called mass spectrometry. Anyway, Hans Gay and colleagues demonstrated that certainly in pheochromocytomas, you had this associated with SDHB mutations, you had a very dramatic rise in succinate. That's been well known for a while. But what was new data is that there was also elevations in other metabolites, one of which he focused on was something called spermine, which is part of something called the polyamine pathway. Why is that important? Well, again, I'll come back to that idea I suggested to you at the beginning of the talk. It's easier to make an inhibitor than it is to replace something that's missing. So we haven't got gene therapy to replace missing SDHB, but we have got compounds that might inhibit some of these metabolites. So elevated spermine can be blocked by using various compounds that have been studied in other cancers, such as neuroblastoma, for decades. And these agents, at least in this preliminary study, and I should emphasize that this was in cell culture and in animal models, not in human beings, but there was some promising results using these inhibitors that seem to block specifically pheochromocytomas associated with mutations in SDHB. So there's a lot of profit in studying these tissue samples, because you understand how elevations in various cancer molecules might be specifically targeted with therapies that may have been sitting on the shelf in cancer medicine for some time. I'm sure we'll hear a lot more about this. It's a very interesting and exciting discovery. So agenda item number 2 was developing new targeted therapies, and there's been admittedly very slow progress in this due to the lack of preclinical models. The disease is very heterogeneous. It's also rare, so that means patients with metastatic pheochromocytoma tend to be widely scattered in geography, and that makes the conduct of standard clinical trials quite difficult. You understand that most clinical trials are usually conducted in some targeted centres, what we call multi-centre trials, but those centres are high throughput, and so you get they are able to concentrate a large volume of patients to study cancer medicines. With this disease, because patients are so widely scattered across the globe, it makes it a lot harder to get in touch with expert centres that have the capacity to do properly designed cancer trials. But there have been a few advances, one of which was asked specifically by, on the chat, the questions. So I'm going to talk about that. Certainly Azedra, high specific activity MIBG, seems to be the real deal. There's been, I think, very good evidence to suggest that Azedra should be considered for the treatment of patients with metastatic pheochromocytoma. It's got good evidence for tumour effect, and it also reduces the excess catecholamines associated with these tumours. And I think my friends, colleagues, and patients in America are very lucky to have access to Azedra. For me in Australia, I can't get Azedra, although I'd love to get hold of it. So what we've been studying is a different compound called, which you will know as Lutathera, but which I will call lutate, because that's what we have access to in Australia. So in talking about these radionucleotides, I do want to, again, this is, I'm sure, common knowledge to you all, but pheochromocytomas are adrenal tumours. So the adrenal gland sits on top of both kidneys. and so the pheochromocytomas will affect one or both of the adrenal glands. Paragangliomas come in two flavors. They can either occur in the thorax or abdomen, what we call thoraco-abdominal paragangliomas. So in the thorax, paragangliomas can be in the mediastinum or the lung or the heart, and in the abdomen, they tend to affect the sympathetic chain, which runs either side of the aorta. And one very well-known paraganglia is called the organ of Zuckerkandl, which sits just at the bifurcation of the aorta down in the pelvis. So, but head and neck paragangliomas affect, although they share many features of pheochromocytomas, they are quite distinct tumours in the way they behave. One of the distinctions is that they rarely secrete adrenaline, and the other distinction is they're much less likely to kill people. So if you've got a head and neck paraganglioma, even if it becomes metastatic, it appears to be quite an indolent disease. In head and neck paragangliomas, the most common one is the carotid body tumour. You can see just a little faint lump here in this lady's neck. That's a carotid body tumour. And here's a paraganglioma sitting behind an eardrum, which is another location you can get a head and neck paraganglioma. I guess the most troublesome head and neck paragangliomas are the ones that occur in the base of skull, what we call the jugular paragangliomas, and that's because they can do quite a bit of mischief even when they're quite small. Carotid body tumour can grow. Sorry, a carotid body tumour doesn't have much which it can cause mischief if it grows to a few centimetres, whereas in the base of skull, tumours that grow even by millimetres can cause a lot of mischief. So, as I said, I don't think there's much doubt that AZEDRA is the real deal for treating pheochromocytomas, tumours of the adrenal medulla. And sorry, Eli, I'm going on a bit. I won't be very much longer, I promise. AZEDRA may not be as effective for head and neck paragangliomas. So what does that mean we have to offer patients with head and neck paragangliomas? Well, Lutathera or lutate, I think is beginning to show some promise. So this, it's been, Lutathera has now been, or lutate has been studied in a number of different countries now. And I've shown some data from my colleague Rod Hicks in Melbourne. He published a few years ago his case series on the use of lutate in patients with metastatic pheochromocytoma, but also head and neck paragangliomas. And here on the slide, you can see a patient with a head and neck paraganglioma, quite a large one. It was really disfiguring and causing a lot of discomfort. It's also quite widely metastatic. And you'll see that after just 2 cycles of lutate, just the dramatic response of this tumor. I have to say that this is an extraordinary response and certainly not the general experience. But accumulating data from across the world, and here's another paper from Germany that had treated a number of head and neck paragangliomas with lutate. And after treatment, all had stable disease. And I think this is the general experience of expert centres using lutate, that if you give lutate, you can get tumours to stabilise so they no longer grow. Some have reduced tumour, so 4 out of the 7, and it's a bit difficult to see, but if you see dropping of these lines below this line here, dropping of the bars below this line, it means the tumour shrank. So this tumour, this tumour, this tumour, and this tumour all shrank on lutate. And I'd suggest that lutate might be suitable for head and neck paragangliomas that are refractory or unsuitable to standard techniques such as surgery or radiosurgery. One of the things about lutate is it appears to be extremely safe. It's got very little impact on the bone marrow or on the kidney. And unlike surgery, it's not a disfiguring treatment. It doesn't leave people with scars. So it's certainly in the mix now. I know that the NIH is doing a properly conducted clinical trial for Lutathera in paragangliomas, but I believe that we'll see a lot more about the application of lutate in the treatment of paragangliomas in the near future. So my final agenda, and this won't take very long, but it's a real passion of mine because it involves the patient group. And so this is where we all get to connect together. It actually has some analogies with what we're going through with COVID at the moment. You know, one of the most important defenses of COVID is contact tracing. If we can identify who's got the virus, we can help prevent the spread of the virus to the rest of the community. But we can also offer early treatment to patients. And that's exactly the analogy that I want to draw for paraganglioma. Because it's so commonly hereditary, If we do contact tracing, which in this scenario means family tracing, then we have the ability to pick up these tumours early, and actually when they're small, they can be cured with surgery. It's only once they've got large that they become inoperable, and we have to reach for the cancer medicine war chest. So we've known for a long time that tumour size is strongly related to the risk of metastasis. And if we detect these tumours early, it gives the best possible chance of cure. And I'll underline the word cure. So what we need to do, and this is in partnership, is really reach out to spread the message that if you have a paraganglioma or a pheochromocytoma, get tested, go and see genetic counselor, have your genetic testing, because if you are carrying one of these hereditary ticking time bombs, it makes a huge difference to your family members, who can then get onto a proactive screening program. And there's various opinions about what that screening program might be. There's general agreement across international guidelines that if you're carrying a genetic mutation, you should have an annual test for catecholamines. And then there's some debate about the intensity of screening, but what we do here in Australia is 2 yearly whole body MRI imaging and or gallium dotatate imaging, which seems to be quite sensitive. So as I say, hereditary paraganglioma syndromes are due to mutations in a number of different genes, and it offers us the real ability to offer bespoke care, so tailored information about personal risk and the early identification of tumours. These rare syndromes are more common than we expected. This paper that we published a couple of years ago was referenced at last year's meeting, but I just want to emphasise that we did an analysis looking for mutations that we knew were associated with pheochromocytoma, and we looked in population databases, which are now available through the Broad Institute. And one of these population databases, which is now commonly used, is called NOMAD. We did some mathematical calculations to suggest that maybe 4,000 Australians have hereditary Pheo gene mutations, which was many more than we expected. And just to extrapolate to the American context, that would be 40,000 Americans. So you're not alone. If you're carrying one of these genetic mutations, there's more of you out there. And if we identify these patients who may be carrying the genes, it gives us the opportunity to find the tumours before they turn out and cause mischief by metastatic disease. And just the emphasis about how important genetic screening is was made by the French group and Paul Jimenez-Roqueplo, saying that if you didn't do genetic screening, you had the tumours could grow and metastasize, compared to if you did do genetic screening and picked up the tumours early, you could get the tumours before they metastasize, and they did a lot better. So there's the logistic problem. How do we reach out to patients? How do we spread the message? So this is where I think organisations like the Pheo Para Alliance are really important. It's about reaching out. It's about creating networks. It's about connecting patients and their families with expert centres that have a passion about this disease and want to help people navigate this difficult journey. And I think one of the things that we will see in the very near future are real links through doctor trackers to allow patients to link with centres that have a real interest in their personal journey. So just to conclude, I've taken you through 3 of my personal research agendas. There's so much more we could talk about, and I apologise for only focusing on 3, but we've spoken about hypoxia-inducible factors and a possible therapy against HIF that's in clinical development. We've spoken about telomeres that keep the ends of cancer chromosomes from falling apart. We've spoken about therapy development, and there are quite a few therapies, and what we do need to do is try and connect patients with these therapies to centres that are willing to put them in clinical trials. And we've spoken about detecting paragangliomas early, which is feasible in hereditary disease, and we need patient engagement in screening protocols. So I just wanted to shout out to my lab, all who work extremely hard in Pheo research, and I know that they want you to know that they're working hard on your behalf, in particular Trish Dwight in my laboratory, and a pathologist I've done a lot of collaboration with, Anthony Gill. I've got a number of PhD students working on pheochromocytoma, including Dalia Davidoff and Amanda Seabrook. I also want to specifically thank Richard Tottel and Carol Pachak for their generous collaboration and friendship over the years. And I'll take questions.
Thank you, Dr. Clifton-Bly. That was amazing. We really appreciate it. The research for this is ongoing, and as he mentioned, the scarcity is such a problem to where finding tissue work to work on any of this is always going to be a problem if we don't have more of it. So we do have a notation here that I would like to address. If any of you guys are going into surgery or anything coming up soon, we'd like to ask for tumor tissue donation. If you have an upcoming surgery, you can participate in the Pheo para research by donating your tumor tissue. It's fast and easy, and the consent is at pattern.org, and they will take care of the rest. Your tumor tissue will be studied to find new treatments. So I've got one more announcement, but I'll make that at the end. I'll go ahead and start with the questions.
I was just going to say that that's such an important initiative, the Tumor Project. I'm so thrilled that it's happening. I know my patients in Australia would love to be part of it. It's going to be problematic with the logistics of tumor transport, but we have tissue banks that we've been using for 30 years here in Australia. It really does create the platform for discovery. And I know in particular the Pheo Para Alliance is working with the Broad Institute, and as well as other research institutes across the country. This will be game-changing. And I think I just have to congratulate the Pheo Para Alliance on what is an outstanding initiative.
Thank you. And thank you to anyone who is considerate enough to donate. Okay, first question up. Does genetic mutation have any determination on whether MIBG or Lutathera is effective for metastatic disease?
It's a really good question. We don't know the answer just yet. We're beginning to try to understand that. So I don't have any simple answer for you today, but it's an excellent question and one that is at the forefront of this data acquisition. The data is coming in, and really that's one of the most important questions. And I'll just illustrate how important it is. We know that SDHB tumors have some response to a chemotherapeutic agent called temozolomide. Mm-hmm. We know that SDHB tumors have some response to lutate. It may be that Lutathera needs additional what we call radiosensitizing agents, such as temozolomide, for its full effect. So these are really, this is clinical trial and evolution, if you like. We're studying the response of tumors based on their underlying genetics, and then trying to work out in real time how we can improve the effect. So excellent question. I wish I could give you a straight answer, but it's right up there. You're on the money.
Thank you. Next question that just came in. Is there any new research on treatment of PGL bone mets?
Another really good question. I like that question because, as I'm sure your questioner understands, a lot of the traditional chemotherapies actually poorly target bone metastases. And a lot of that has to do with how bone actually protects the metastasis from taking up cancer agents. Turns out that the radionuclides, such as MIBG or Lutathera, actually do target bone metastases quite well. So the bone doesn't act as a shield for the metastasis in the same way that it seems to shield the metastasis from chemotherapies. The other treatment, though, and this is really treatment that's come out of breast cancer and prostate cancer work, both cancers of which frequently metastasize to bone, and that's using commonly available osteoporosis therapies, such as zoledronic acid, which in my country is called Aclasta, or denosumab, which in my country is called Xgeva. I think it might be called Xgeva in America as well. So these osteoporosis therapies, which are used throughout the world to prevent fractures, are actually very helpful to prevent cancer growth in bone as well. And we've seen the scarcity of pheochromocytoma means that the data is not as solid as it is for breast cancer and prostate cancer, but there's a real genuine belief that these osteoporosis therapies can help people with pheochromocytoma that's metastatic to bone.
Do they alter those kind of therapies at all, like by making a chimera with a chemotherapeutic agent attached to it, or do they?
I love that question. I love it, absolutely. Because you can target stuff to bone. You can do it. And we've, in other bone disorders, so as Eli, as you mentioned at the beginning, I have twin interests in metabolic bone disease. You can bring therapy to bone using these osteoporosis drugs. So because the osteoporosis drugs bind to bone, you can make them into homing missiles and bring a therapy to bone. I think it's a really good idea. I confess I'm not doing it. I'd love to do it. But again, I think that's a spectacular idea. Definitely worth thinking about.
Okay. The next question, I think you answered towards the end of your presentation. This person asks, I didn't have genetic testing when I was diagnosed. Should everyone get genetic testing?
My answer to that is yes. There is some debate. Is there an upper age limit? One of the largest families of pheochromocytoma that we discovered in Australia, the person who presented first with a pheo was 72 at the age of diagnosis. So I don't think there's an upper age limit, but some of my colleagues think, well, if you've got to the age of 70 and you've got your pheo then maybe you don't need genetic testing. I think the more we discover about this, they are a bit like sleeper cells, these genetic mutations. So some people can go a long time without realizing they've got tumors. So my answer, my preference, my bias, my prejudice is that everybody with a pheo should get genetic testing because it makes a big difference to the rest of their family. Can I also say though that if you are carrying a hereditary gene, it's a real difficult conversation. And you need expert help for that conversation with the rest of your family. So they are can of worms discussions, and there are experts that are specifically trained to help you through that process. Because there's a whole range of emotions associated with hereditary syndromes. There's guilt, there's anxiety, there's fear of the future, there's what happens to my children. And those emotions, we need to take the blame out of it. We need to say that this is an opportunity, not a disaster. We need to look forward and help people navigate this difficult journey. And genetic counsellors are really skilled at these conversations and helping you navigate those difficult discussions that might occur in the rest of your family if you're found to carry one of these hereditary genetic causes of pheochromocytoma.
Okay. Yeah, wonderful. Okay, so the next one is a two-part question. What percentage of SDHB mutations are variants of unknown significance, better known as VUS? And a VUS, I understand, is you find a mutation, it's not necessarily classified in any sort of database, but you know it's not what's normally supposed to be there. Is that correct?
Yeah, that's a good question. So VUSs, VUSs are the bugbears of clinicians. And that they've really just appeared in the last 10 years as we've learned more and more about genetics. My short answer is very few. These days with what we call orthogonal testing, which would either include studying the tumour with special stains, looking for SDHB, something we call immunohistochemistry, or with metabolomics, where we can measure succinate in the tumour, we should be able to resolve whether a genetic mutation is causative or incidental to it in pretty much everybody. So my answer is very few SDHB variants should be labelled VUS at this stage. We've got tools to unravel the identity of those variants. But you make a good point, and that's not every mutation in a gene is the cause of a syndrome. We all carry mutations. A conservative estimate is about 20,000 mutations in our DNA in every single one of us. And so, most of the changes that occur in our DNA are incidental. And working out whether a particular DNA change is causative of disease or not is a really important topic.
Maybe they're just what make us unique, like a thumbprint. So
that's exactly right. It's our genetic thumbprint. You're bang on.
Okay, so the second part to this question is: would it be reasonable to assume that a paraganglioma of the bladder removed at age 17 was caused by the gene mutation? The mutation was also confirmed to be inherited by the mother, although it was never presented with her.
Yes, that's true. So again, this takes us back to the complicated discussion about tumor suppressors. So for the vast majority of genes, we get 2 copies, one from mum, one from dad. And if you've got a hereditary syndrome, one of those copies is switched off. But to get a tumor, you actually need the other copy to be switched off as well. And that's what we call a somatic loss of the second copy. So the tumor suppressor, you inherit one copy, and then by chance you lose the second copy. And it's the by chance bit that we really struggle to understand. And it just seems like cosmic bad luck in the vast majority of cases. There's nothing that we know that causes the loss of the second copy. So it's not that you ate something wrong or you did too much exercise or whatever. It just happens by chance. And when that second copy gets switched off, bang, that's when you get the tumor. But that will be variable in different people at different ages when that second copy is lost. So some people will live their whole lives without the second copy of SDHB being lost, and no tumors will result. Other people, a second copy will be lost in the bladder or the adrenal gland and cause a tumor. So we know that inherited copy is the cause of the tumor, but we know that the other events occur as well that help explain why a parent might not have a tumor and a child may have a tumor.
Right.
It's a complicated discussion. I hope that I've been able to give some clarity to that.
Okay. All right. In a book entitled The Big Five by Dr. Sanjiv Chopra, in the foreword, he mentions there is emerging evidence that a Mediterranean diet is associated with longer telomeres. Would you care to comment?
Yeah, so long telomeres are bad for cancer, but they're good for the rest of us because they're essentially a parallel of good aging. So if you've got long telomeres, then you're going to age well. And that's the concept of the Mediterranean diet being helpful to healthy aging. As I say, that doesn't mean that it will cause long telomeres in cancer. In actual fact, there's quite a bit of research coming out in general cancer biology showing that Mediterranean diets, or to be frank, any good healthy diet, lowers it by lowering insulin levels. It can lower the risk of cancer developing and lower the risk of cancer metastasis. Whether that's true for pheochromocytoma, we don't know. But so the influence of diet and cancer outcomes is not simple. It's not just about telomeres, and it's not just about weight. It's got all sorts of things about how hormones are regulated, but it's a good question. The Mediterranean diet in particular may have benefits on multiple levels. It's certainly beneficial for cardiovascular outcomes and seems to promote a healthy aging concept.
Sure. If you can get past the tzatziki sauce, then you're good.
Right.
Okay. So this next question is one that I've been curious about too. Is there any research investigating whether extremely high secretory pheos will go on to reoccur or metastasize, especially where genetic syndromes couldn't be identified?
It's an extremely good question. So there's been a lot of research on on the factors that will predict who gets metastatic disease. And the 4 main factors are genetics. So in particular, if you've got SDHB.
Right.
Secondly, the size of the tumor. So for a pheochromocytoma, it's above 5 centimeters. The third factor is where the tumor is. So you're more likely to metastasize if it's a paraganglioma, in the chest or abdomen, than a pheochromocytoma. And the fourth factor is secretion. But it's the type of hormone that's secreted, not the amount, that seems to be important. Now, the amount of hormone that's secreted is directly parallel, for the most part, to tumor size. So the amount of how much adrenaline you're secreting will be proportional to how big the tumor is. But very interestingly, tumors that secrete only noradrenaline are much more likely to metastasize than those tumors that secrete adrenaline. So if you've got an adrenaline secreting tumor, it's actually less likely that those tumors will metastasize. And there's one easy explanation for that. Adrenaline is produced by highly differentiated cells of the adrenal gland. So in order to be able to produce adrenaline, so again, sorry for if people are unfamiliar with this concept, we've got two brands of catecholamine. There's noradrenaline and adrenaline. Both of them come from the adrenal gland or from paraganglioma. Paragangliomas mainly secrete noradrenaline. So adrenaline, it requires the cell to be much better differentiated. And if the cell is better differentiated, it's less likely to metastasize. So if a pheochromocytoma is secreting a bucket load of adrenaline, it seems less likely to metastasize.
Right. Interesting. And is that the same as reoccurring as well?
So recurrence depends on the surgeon. So if the surgeon's got the tumor out, then there's a very low risk of recurrence in the adrenal bed. If there's been a bit of tumor left over, if the tumor is big or invading into surrounding structures, if the surgeon can't get the whole of the tumor out, then any tumor can recur regardless of its secretory pattern.
Okay, that makes sense and makes me feel a lot better. Have you experienced patients with both endothelial dysfunction and pheochromocytoma?
I thought that was a really interesting question. So endothelial dysfunction is a hot topic at the moment with coronavirus. So coronavirus appears to have its main effects on the endothelium. And to a large extent, pheochromocytomas and the catecholamines do have direct action on the vasculature. And we know this from the common symptoms like racing heart, sweating, pallor. So adrenaline causes constriction of arteries. And so during a pheo crisis, your hands and face will often go quite pale because of the constriction of arteries. And so I'm sure there is endothelial dysfunction. It's just, to my knowledge, not been very well studied. And whether nitric oxide plays a role in how pheochromocytomas cause the disease it does. So it's a really interesting question. And I might ask the chairman if I could park that question to one side and go and do a bit of research on it myself and come back to you.
Sure, absolutely. Okay, we'll move on to the next question. Quite a few clinical trials on clinicaltrials.gov have a goal to treat pheochromocytomas and neuroblastoma. What is the commonality between the two?
You've got a spectacular audience here, Eli. These questions are so insightful. So we've known for decades that neuroblastoma and pheochromocytoma are essentially cousins. They share some of their genetics, although neuroblastomas are not directly related to succinate dehydrogenase per se. We have seen a few families where neuroblastoma is part of the syndrome. But more in terms of a lot of their other genetic manifestations, the genes that are switched on inside neuroblastomas are very similar to genes that are switched on in pheochromocytomas. And that includes the telomere story that I mentioned to you before. So there's, for sure, pheochromocytomas and neuroblastomas are cousins. That doesn't mean that treatments for neuroblastoma will automatically be helpful for pheochromocytomas and vice versa. But there is a real sense that by talking between the two research communities, so as many of you know, there's a big neuroblastoma research community in the same way that there's a big pheochromocytoma research community. And you may be more surprised to learn that those two fields rarely talk to each other. But we have realized that was a mistake and we need to talk more between each other. So in Australia, in fact, two of the big neuroblastoma laboratories are right down the road from where I work. So I've actually spent a bit of time over the last few years wearing out the shoe leather, walking down to the neuroblastoma laboratories and learning why they do what they do, where they think the new discoveries are going to come. And more often than not, they are relevant to pheochromocytomas.
Interesting. Well, let's hope there's a commonality that they can work together on. Okay, is there research?
I was just going to say one other point on that. And again, it's a sort of interesting reflection on human nature. And that is for the vast majority of hospitals, there's a division between children's hospitals and adult hospitals. That's true in Australia. I believe it's true in the US as well. So that's not to say that some hospitals won't have both, but in general, children's hospitals become super specialized and concentrated in their own facility, distant or at least distinct from the adult hospitals. Pheos tend to occur in adults, neuroblastomas tend to occur in children. So that is why there's been this sort of loss of connectivity between the two research groups. We're sailing on two different boats, and the boats got further and further away from each other.
Yeah, I think that's more common than we care to admit on a lot of different departments.
Right.
You know, I personally, for this next question, I don't know how long MIBG treatment has been going on, but is there any research on the long-term survival of patients with MIBG treatment?
Yeah, quite a bit of research. So MIBG has been around for decades, although there are two brands. There's the low specific activity of MIBG that we have in Australia, and which is used internationally and has been around for decades. The high specific activity MIBG, or AZEDRA, has really only been available for a few years and only in America. So I can't comment about long-term survival from AZEDRA. I'm not sure that data is known because it's really only been around for a few years. But for low specific activity MIBG, if your disease responds, the long-term survival is really very good. MIBG, we need to be a little bit careful about bone marrow effects, but it's a generally safe and well-tolerated therapy. Some of our patients have had multiple courses of MIBG over 20 years, 25 years, without noticeable effects on their bone marrow.
Okay, good. Okay, we got the last question here. If there's no reoccurrence of a tumor, but symptoms and elevated catecholamines are present, is it possible for some kind of post-Pheo syndrome because tumor cells are left over after surgery? And I think you commented on that just a few minutes ago.
So the recurrence of tumors is a real problem for the clinicians, because we know that we can detect recurrence by detecting catecholamines in the blood. And we can do scans, but the scans only have, the resolution of the scans is only so good. Now, my personal opinion is that the PET scanning is really highly sensitive to detection of very small volume recurrences. They're not perfect, but gallium dotatate appears to be extremely useful for the majority of pheochromocytomas.
Right.
But you're right. Some pheochromocytomas may secrete things that we don't yet know much about. And so post-Pheo syndromes may be in part due to secretion of hormones like neuromedullin, adrenomedullin, or other substances that may cause some clinical features that have not been properly studied. The other thing, though, which I think is a really important aspect, which we've only just beginning to scratch the surface of, and that is, if you've had a tumor that secretes a lot of adrenaline and you've had it for a while, your body gets a bit used to having a lot of adrenaline. And then you take that out. And this is something I've seen a lot of. Patients can feel very flat for months because they've suddenly lost something that they've become used to. Does that make sense?
So, I experienced it.
Yeah, so you can feel like you've been unplugged from the electricity mains, and that's a real phenomenon. And you know, it has been poorly studied. I reckon we need to understand much more about that post-Pheo syndrome of the fatigue and the lassitude and feeling that you don't have enough energy to get through the day. There's a lot that we need to appreciate about that, and hopefully work with our patients to help them through that.
Right. Yeah. I myself experienced, you know, the gradual calmness, you know, as the months went on. It was fatigue mostly in the beginning, and I think that was mostly due to the severity of the surgery. But I recall by December of 2019, I think I was feeling the calmest and happiest I can ever remember myself feeling. And then 2020 came along and you start getting bombarded with all this information on
Mhm.
COVID and, all the stuff going on in America with the protests and everything. There's just a lot of negative information. So I found myself having some of those familiar thoughts and feelings that I did when I had the cancer, and I kept thinking, oh my gosh, is it back? And, of course, every year I go for my checkups and do my catecholamine tests and my scans, and, I got a clean bill of health, but it just makes you realize that there's some sort of integration back into some kind of normalcy, the
Right.
feelings and the hormones that everybody experiences on a normal basis,
You're so right.
Yeah.
That's amazing. What a story.
Yeah. So, okay, so that was the last question, and I do have another announcement for the Pheo Para Center program. For those of you looking for an excellent team or physician in your area, the Pheo Para Alliance is finalizing the Pheo Para Center program. This is a program where patients will be able to connect with institutions in their area which provide them with quality Pheo Para care. The program will launch in the fall, and we hope to announce the centers by the end of this year. In the meantime, you can use our guide to find the Pheo Para doc in your area. The resource is at pheopara.org under the resources tab, or you can also reach out to Stephanie, who's just kind of the glue behind all of our Pheo Para Alliance. And her email is simple. It's Stephanie with an IE at the end, Stephanie@pheopara.org. So with that being said, I'd like to thank you, Doc, for your time and your dedication to our patients, the community, and the research, and all the wealth of knowledge that you shared with us today. And I'd also like to give a special thanks to Progenics for making this webinar series possible. Join us again at the end of this month. We're having 2 more patient education webinars. So the links to register are at our website at pheopara.org. To sign up for our e-news. So that way you'll get all the information as it's coming at you. Anytime events or webinars like these are coming up, you'll be notified first come as soon as you register. The webinar will be available in a few days, this particular one that we just did, on YouTube and on the website. And we'll also transcribe the Q&A portion, and we'll have that available as well. So that way you can go ahead and print it off. If you have any questions you want to share with your doctor, please feel free to do so. If this information has been useful to you and you're interested in donating, you can do so at pheopara.org/donate. All of your donations go towards events such as these and other helpful resources that we have to offer the community, which with a scarce disease, every bit helps. Thank you for everyone who attended today, and together we're stronger united. Thank you.
See you.